So thank you so much for joining this afternoon. This is the Community Connect webinar series. This webinar series is essentially an opportunity for us to bring different members of the community and the local and global community to discuss specific topics that we think are imperative to the community and are opportunities for the community to learn something new and something important within microscopy. So today's topic is an introduction to Fiji. It's essentially about a 40-minute presentation where Emanuele will tell us about essentially everything you need to know to start making use of Fiji. This presentation is going to be done by Emanuele Martini. Emanuele is a research fellow studying cell migration in oncology and is an image analyst at the AIRC Institute of Molecular Oncology at the University of Milan. And Emanuele is also a very active ABIC member in the Training and Education Working Group, who is currently based in Uganda. Emanuele also spearheaded the in-person Fiji course in Uganda a couple of months ago. So I think he's the perfect person to tell us about this topic today. So Emanuele, without further ado.
Okay, good afternoon, everybody. So thank you, Precious, for the introduction. It will be a quick course about Fiji, and a little bit, I will use also Fiji to remind some bioimage analysis concept that is always useful. And I think, okay, now I see myself, I think I can share. Okay, thank you, Precious. Okay, let me reduce the people. How can I do it? Okay, okay, I'm fine. Okay, so first of all, why Fiji? Fiji is a very extremely widely used image processing software in the community of microscopy and scientific image analysis. Is a good tool because it comes with a lot of ready-to-use plugins and pipelines to do facilitate scientific image analysis. The main reason why it's used is because it has a quite user-friendly interface. Moreover, if you compare it to those kind of more command line programs, it's basically a point and click software. So you load your images and you click on buttons to perform analysis and visualization steps. And then it's another very important stuff is that it supports almost all the images coming from the bioformats. Bioformats are those ones coming from microscope, specific file formats coming from microscope, and it's able to interpret them in many ways and in a good way for scientific purposes. It's able to also understand and visualize and analyze well multiparametric acquisition. With multiparametric acquisition, we mean those ones that is multi-channel, multi-time points, like a time-lapse, a movie following live cells, or multifocal planes when you want to reconstruct a 3D of your cells, of your nuclei, and something like that. Then for sure it's also quite important that it's free and open source. So this also helps a lot to be used in the community. Then it has a very important worldwide community. It is a website where you can really ask and you will be answered by many very good experts in the field in a few days. So use it a lot, not only for Fiji, but Fiji is one of the main part of the worldwide community of image biological analysis. Then for the people that are more on computer science part, and less in the biology part, it's also quite fast. It's not the fastest software in the world, but it's quite fast. And it's also, sorry, okay, and it's also automatable and expandable with different programming languages. You can write your own plugin, your own analysis in Java, in Python, in Bean Shell, and in many other languages, and then you can put directly inside the Fiji software and use it for your own analysis and purposes of your experiment. So in these 45 minutes, I hope to be able to show you how it works, a classical framework in the biological image analysis world. So I put it in red because it's very important. You always start with your biological problem. This is your research project why and what you are interested to see in an image and why you are using microscopy to reply to your to answer to your biological problem. Then it will be splitted in two parts. One about visualization. It seems not important part, but a lot of time is the most important part to understand what are you seeing, if you are seeing it well, if you are watching your images in the proper way, and so on. And then there's another part at the end about the analysis, so how you use the Fiji to do some analysis of your images, so extract some data and something like that. So before that, I just wanted to start from the raw data. What are raw data? The one that you get from the microscope. And in our case, they are the pixel, no? So you get your image and you get the pixel. What is a digital image for what we are interested in these 45 minutes and in the digital image field? It's the result of two digitizations. One is called sampling, okay? And it's related to the coordinates of the image. So how many pixels you have, okay? In how many pixels you are able to divide your object, your field, your slide, your object you are acquiring with the microscope. And the other one is related to the signal, and it's called quantization. And is related to the intensity of what you are seeing. Okay, so just to put some few slides about it to be sure that we are all in the on the same page about what we are saying when we talk about images in the digital field, so digital images. So the first digitization is the sampling. So the sampling is simply get pixel in the image size width and depth and sample your original image in a digital space. So for each point, you have a couple of coordinates in X and Y telling you where you are in the image. So then I also use this guide to at least have an idea that when you work with biology, you have usually two kinds of acquisition. One is the grayscale, so you have only one value for each couple of coordinates of your matrix of points called pixels from the sampling. The other one are when you acquire colored images. So instead of having only one value of intensity for each couple of coordinates, for each coordinates in the sampling space, you will have a triplet. That is, in this case, the red intensity, the green intensity, the blue intensity, the so-called RGB image. But from where these values come from? These values come from the so-called quantization, the other digitization we talked about. So the quantization is in how many levels you want to divide your original signal. So here is 1D because I think it's a little bit easier at the beginning to understand instead of going directly to the two-dimension of an image. You can imagine that your real signal, your real-time series is like something like this red one, that it has all the values possible in this range. When you use a certain bit depth, what you are saying is that you are dividing your original signal, your real signal in some digits, no? So in some levels. If you have only 2 bits, you can divide it in 0, 0, 0, 1, 1, 0, and 1, 1. So in only 4 levels. Increasing the bit depth, so you are increasing the discrimination power. And most of the time, when you work with images, you will have something like 8-bit or 12-bit or 10-bit. So it means that you have from 0 to 255 values of gray where you can discriminate. Of course, if you increase the discrimination power, you increase the bit depth, you will be more able to see differences in terms of intensity and in terms of morphology of your biological samples. One very important thing of image analysis, the image histogram. What is the image histogram? The image histogram is the distribution of all the intensities in your image. So how many pixels of a certain brightness you have in your pixel. By convention, the pure black is put to 0 and the pure white is put to 2 to the power of the bit depth minus 1. So if you have 8-bit, it will be 255. And then you scale, your intensity goes from black, darker, and then less darker up to white. It is very important because you use it a lot in both in visualization and in analysis. One of the main topics to understand when you want to do image analysis. So from here, I will start to use a little bit Fiji to see you how it's easy to use. I will do sometimes with this guy, sometimes directly with Fiji because we have only 40 minutes, so I cannot do everything by using the software. But I just want to use to show you how easy it is with Fiji to do some basic operation and then some more complicated operation. What we will see in this first part is about the visualization. So we will start with the simplest acquisition, so just a single image, and then we will increase in acquisition complexity, going to have more channels, more time points, and then even to 3D acquisition. So of course, to open your image, you can just drag and drop your image. Let me open one image from here. And when you drag and drop, your image is open. What you have already in this image, let me just zoom it out a little bit like this. Okay. What you have in this image? What you have in this image is the main information. No? So here, as you can see also in this guide, you have the bit depth, so the quantization bit depth and the sampling. So how many sampling you have? No, you have. It was acquired with 16-bit, and so you have 2 to the power of 16-bit levels of gray, and then you have 1000 by 1000 pixel as a sampling. Then if you move with your mouse, you can go and investigate the value of the image in that point. As you can see it in the toolbar. So if you see when I move around, the value in the toolbar here will change. It will change, no? So when I'm a little bit in the black area, I will expect a lower value. When I go to a bright point, let's see if I can go to this bright point here, you will see a very much higher value, no? Because there is more signal, there is more intensity, and as we said, as long as you go more to the white area of the histogram, you will see a higher value. Okay, so this is just to show you that the main information are already displayed for you when you open the image, and the main information is the one we say. So bit depth, image size, pixel intensity. Of course, you can extract the histogram of the image, so you can understand how is the distribution of your pixel just clicking on histogram. As you can see, let me see, okay, here you can see better, you have a lot of statistical values, and so you have the minimum intensity present in your image, the maximum value intensity present in your image, the mean intensity, and then here you can see how it's the intensities are distributed on your image. So for example, from an histogram like that, what you can already estimate is the background is around this value, no? Because it's the most present value, so it's the mode, so it's 168 values as intensity. You can see that it's extremely present here, and then this is more or less the background of your image. So if you want to estimate in a very qualitative way, in a very basic way, the background of your image, the histogram is already enough, and it can tell you something, and you can use it as a background removal from in your image and in your analysis. Now we just continue to have a little bit more complicated acquisition. This acquisition, for example, is an acquisition of figure cells or mammal cells. You have marked, I don't know how know about microscopy in general and about fluorescence microscopy, but for the scope of this course is just enough to know that you can put some marker and then you can look at it in different visible wavelength spectrum. So you can mark the nuclei with DAPI, and then it will show you, for example, in the blue wavelength, you can put some markers for microtubules, and then you can see it in the green spectrum. And then you can put maybe some antibodies, some markers for centromers, and then you can see it in the red spectrum. One thing to know is that most of the time, to do this kind of acquisition with fluorescence microscopy, what you use is the black and white camera. So When you then open it after acquisition at the microscope, you expect something like this. Let me find the images. Let me put all of them. You can multiple select and then just drag and drop. As you can see, these images are not in the color you acquire them, in the spectra you acquire them. Because this is for Fiji a black and white image, okay? So for them, it's just a single grayscale image. But a lot of time you really want to put the color to visualize them better, to show the people which kind of markers you used and something like that. To do that, what you use is the so-called lookup table. Lookup table just means that you change the visualization, so not the really values of your image, but just how you are visualizing them to different kind of colors, okay? Usually the basic use of the lookup table is just put for the nuclei that you acquired in the blue channel, you can put a blue lookup table. For the green one, you can put a green lookup table. Let me find it. And then for the red, since you acquired in the red channel, you can put a red lookup table. This thing, it seems quite useless, but it becomes more useful if I show you that you can put all together, and you can put all together doing a merge channel. So you can merge them all together. So you put in the red, let me find it, in the green, this one, and in the blue one, the W. Let me find it. What you have now is it's a so-called stack. Let me go here. So what is a stack? A stack is an image set of multiple spatially or temporally related images in a single window. Why, for us, this is a stack? Because if you imagine what we have done with the microscope, we acquired the same field, so it's spatially related. So it's the same field of your slide of your cells. You acquire the same part of your sample in the same place, so spatially related, but with different color. Okay, so here you have a stack. Please note that some terms I'm using, like lookup tables, stacks, and something like that, are in Fiji, but they are common technical terms for image analysis. So it's nice also to use Fiji to show you, and when you maybe go to another tool, you will still find lookup tables, you will still find images stack and something like that, because they are common names for technical terms. So what you can see here, I will show you in, I can crop just a piece of it. Let me crop this one. What I have at the moment, it's a stack. So in principle, this is not a color image, a colored image, okay? It's like, they are still three different images, just overlaid one in a single window. For example, if you want, you still have the histogram. If you extract the histogram for them, you will extract the histogram of the red channel, then I can move to the second channel, the green one, and I can extract the histogram of the second channel. So they are still single images. If you want then to send to someone and show them these beautiful three cells, you have to convert it to RGB, or you want to put in a presentation or somewhere. And now if you see in the toolbar, if I move around, you will see three values for each point where I am with the mouse. No, if you can check here. Here now is no more a stack. If I extract an histogram of this image, let me extract it. You can see that is now a single is no more divided in green, red, and blue. Instead, here if I can still move around and can still evaluate, see, and watch and see values of as a single image. Okay. Very important. I didn't stretch it a lot. When you apply lookup tables, like as we done before, is one of these operations where you are not really changing the values of your pixel. You are just showing them differently, just because you want to, in this case, to put them all together or for other reason. it's like you cannot come back when you apply a lookup table like this because you are not changing the real values of your pixel. Now, we also have sometimes we also have temporally related experiment, like a time lapse. This is an experiment I just explained very fast. It's a so-called wound healing experiment. You grow your cell up to a certain level, then you scratch them in the middle, and then you want to see which condition closes the wound, the hole, faster than the other one. So it's a cell migration assay. What you can do, just from a qualitative point of view, let me find them. You can open them like this one. Now I forgot to put another one. Sorry, and then you can watch it as a movie. And then if you have two of them, you can combine them and then let them go together. And when you see together all of them, you can at least qualitatively compare them as seeing that, for example, the treatment is increasing the speed of closure. So here, it's nice to see that you have some tools that are thought up for biological experiment, like this one to have with just a click to combine two movies and then play them together. If you imagine, if you don't have that, finding some other tools in generic purpose image, I don't know, like PowerPoint, like Paint, like Preview on Mac, those tools are not thought to do this kind of stuff. Here, with just a click, you are able to do this because Fiji is taught for biological image samples. So they know that a lot of time you have to compare time lapse. Another things you can do, and I will don't show you in Fiji, but I just want to show here, is the so-called Z stack. So what is a Z stack or a volume reconstruction? It's when you acquire on the same space, or so on the same space on your biological sample at a different Z, so a different focal planes, and then you can do it with a confocal microscope. In this case, is a East acquisition. So what you can see is a different plane, so a different height in space, different. How is your East? What you can do with this is many things. I don't want to go too much into the details because we don't have so much time, but I just want to show that you can reconstruct in 3D. So this is a 3D reconstruction of your East acquisition coming from all those slices you acquired at different height. And these things, of course, Fish is not the best tool to do 3D rendering. There are many more powerful and nicer tools, but it's very nice that it allows you to do it with just a simple click. I didn't do that today. I will not show you in Fish directly, but just imagine that it's just like a single click, and you will produce a very quick 3D rendering. Then, if you want something better, you can go to better tools. But at least you have already a 3D rendering tool in your in an open and free source and free software. Another thing important that you can do and you have to do in biology is to always consider that your biological sample is something physical. So before we were seeing pixel, but now we can we really want to know how big in terms of physical unit is your, I don't know, your nuclei, your cell. So you need to calibrate your image. There are I just want to show you two ways to do it, okay? Sometimes you don't have the so-called metadata. What are the metadata? Metadata are files, image files, bioformats images that include information about how your image is acquired. So in this case, if we have metadata, we could have already the size of your image in terms of microns. If you don't have that, there are different ways to calibrate your image. In this case, this is the formula, but this is not so much important. It's just important to note that if you don't have your metadata, it's you by yourself that you have to calibrate your image. What it means, calibrate your image? Calibrate your image, it means telling to Fiji how big is in microns your pixel. So here you are defining that one pixel is length 0.32 microns. When you do that, what you will see is that your image now, let me increase this one for a while, reduce this one. Let me put this on front. What you will see here is that you have still the size in pixel, okay? So your image is still, Fiji will still provide for you the real pixel size, but then it will also provide for you how big is in microns, so in physical unit, your image. That way you can, in principle, compare different sizes, different images, even acquire with different microscope, because you will be able to calibrate to the real physical micron space. And then you, of course, you can put a scale bar, okay, in width. So here you can put on your image that this length is 106 micrometers. What I want to show you is like when you have instead the so-called bioformats. For example, this one, let me go to open it for you. Let me find it. This kind of microscope provide for you a .lai file. Of course, .lai file, you are not able to open it with normal image softwares. It's a bioformat. It means it's a format created by a microscope that you can directly open in Fiji. So you do this, okay? It understands for you that is a bioformat, and then you can open it. You give it a little bit of time. Okay, it's reading all the metadata. This is a metadata file, and then this file is organized like this. Okay, it has different channels. It has so many channels, and everything is already organized for you. Imagine if you don't have a metadata like this, a file like this, to open all the single files, and then yourself try to organize this kind of acquisition. This is also metadata, an hyperstack, sorry. It's an hyperstack because you have different Z, so you acquired to do a 3D reconstruction, for example, but you also have different channels. So, for example, you have the actin, the centromers, I think there are the nuclei somewhere, the nuclei here, okay? So, and then it's spatially related, so it's an image stack, but it's an hyperstack because it has two kind of relationship. One is the channel relation, and the other one is the focal plane relation for the 3D. Okay, and then just to be, if you see, this image comes directly. I didn't have to do any kind of by myself to set for them the calibration. Since it knows everything about the acquisition, it also knows how big is a pixel, and so the image when I open it is already calibrated. Okay, I don't have to remember the parameters of the acquisition. It's already inside the method. This is why I invented bioformats, to have a way to consider all the acquisitions, so all the parameters of the acquisition, how many channel, which kind of wavelengths, which kind of Z step you used, the calibration in microns, and so on. Okay, so this was a very quick, but I think more or less covering the whole scenario of microscopy biological images in fluorescence, moreover. So multi-channels, multi-time points, multi-parameter. There's a chat, sorry. Yes, of course. Yes, yes, we can get the video and PowerPoint of the presentation, no problem at all. Okay, so now we will do a step ahead and we will talk about how we can analyze our experiment. So first of all, I want to introduce you the pre-processing step. The pre-processing step, as you can see, is in visualization also. So what is a pre-processing step? In this case, we will see very fast only two of them, the point operation, the neighborhood operation. These ones are used to better visualize, to better analyze, to do denoising. So you want to pre-process your image or to see it better or to have a better way to analyze it. or to do some denoising if you want to remove the background and the noise of your image, okay? So the main two, how can I say, the main two areas are the point operation, the neighborhood operation. The point operation are based only on the pixel intensity value of a single pixel. The neighborhood operation instead, they always consider the intensity, but they also consider the a region around the pixel. So the neighborhood region around each pixel, and not only the single pixel intensity value. We will try to do a little bit more in the details of it in these 5 minutes. The most important one is called the histogram stretching. The histogram stretching is what you also do with your remote control on the TV when you increase the contrast, okay? So, but what it really does, I just put a really basic formula just to show you a little bit of the math behind it. I think it also helps to understand better. What it does is to transform each value of your input image, usually indicated as f, so the value of the original image in the xy coordinate, it will be put it back on the transformed image, on the stretched image, with another value, okay, with a linear transformation. What is the clipping? The clipping is that you are setting also some value above where you put to the maximum of your image, so to 255 if you have an 8-bit image. And also you set a lower level where you want to see only black pixel. Let's see what it means with a real image. Before of it, we want to see why it works. It works because of this. What's happening when you apply something like this is to transform your histogram in something like this. So imagine this is your original image, okay? Original image histogram. So here you are working, your full scale is between 0 and 255. But because you are acquiring real things, so you are using a microscope and you are getting back some values, your image is instead only between 84 and 150 values. What you see is that the difference in grayscale between these two peaks is just here, no? You can let me copy and paste it, and it's like just here. Okay, when you apply this histogram stretching operation, so you apply this kind of algorithm, what you do is stretch the histogram for real, so your histogram become elongated, stretched, and now the difference between the pixel here and the pixel here in terms of intensity are much higher. So for your eyes, it will be easier to watch differences between in this stretched image respect to this one, because this gray level is dark and this one is a little bit more gray, but and the difference is much larger, and your eyes, of course, they will see it in a larger, better, no? They will see the contrast enhanced. This is why it works, okay? And it's really the same things you are doing with your remote control when you do the contrast, when you move brightness and contrast on your image, on your TV. Why it could be useful? This one I want to go back to Fiji because it helps me a little bit to explain this concept a little bit better. Let me go back to the single image, this one. So here is the same image we saw before, okay? And from here, what you can see is that the centromeres are still there, okay, it's fine, but maybe they are not those dots, the centromeres are those dots, they are not so much bright, and then maybe you want to see better, and you can open this tool that is called brightness and contrast because it changes the brightness and contrast. And what you do is really like this, no? I am applying this linear stretching, this is the real same thing we saw before in the formula and the straight line. And so now what I can see, this is the histogram. If I move this one, I'm saying to clip out all the values below the value, this minimum value. So all the values below these ones are shown as black, okay? So it can help, for example, to just enhance your dots that are centromeres. If you have to count how many centromeres you have in your cell, doing some process like this, it can be also useful in the analysis part to count them, but it's also useful to better visualize it. Okay. So this is what's happening. Another thing very useful for it is the qualitatively compare different conditions. When you open two images in an image in Fiji, most of the time the software enhance for you the contrast by itself in an automatic way. To be sure that you are seeing it with the same histogram stretching algorithm, you have to be sure of it. You can do it by setting it for all the images. And this is a case that I want to show you. This image was acquired with a lot of light, so for 1,000 milliseconds, this one was acquired with 100 milliseconds with less light. So you expect that one is darker than the other one because it has received less light. Out of it, you can instead, if you want to be sure that you're seeing it with the same contrast, you have to watch it with the same contrast. So let me go a little bit faster now, because this is, I think, the most important part. There is, of course, non-linear mapping function, so you can emphasize different pixels in different ways if depending on the intensity values. Then there is also the other pre-processing is the one called image filtering, when you want to change pixel based on the neighborhood. I don't want to enter too much, but these are to reduce noise, emphasize, and suppress specific features. One very good use of it is the edge detection. So you can extract the edge of the images of one image using this kind of image filtering. Then there are also some kind of image filtering that is like the median filter to remove some noise. You can use it to remove noise. Okay, it is what we want to show you. Now we will just go fast in last 15 minutes to do some analysis. I will go directly with the presentation because I don't have time to go inside the Fiji tool, but just it's really, for real, it's clicking buttons and you will already be able to do some analysis. Here, for example, if you want to plot the distance and find the distance between these two kinetocores in the east, you can draw a line and then you can plot the profile and then you will see. You can already estimate the distance between them because you can go here and see, okay, there is 100, more or less 5 microns of distance, okay? Another things you want to a lot of time doing is measure morphological and intensity of some object. You can do by hand, of course. Here you can draw your nuclei and then you can set your measurement and you can obtain the result and then store it in your role manager, that is the manager of all the region of interest you have drawn. But then, of course, you can do in a more automatic way, automated way. So one way is to do segmentation. The segmentation is really the process to subdivide an image into subregion or object of interest, in our case, nuclei. There are different ways to do that. The one we will see today is the histogram thresholding. So you separate images based on gray level histogram distribution. There are also other one. One is region growing, split and melt. It's a very big field. And then, of course, there is also machine learning, deep learning, and artificial intelligence that you can use to do this kind of analysis. So it's the I really talked a lot about histogram because it's important in different stuff. And then here it's when you use the histogram to identify nuclei. Okay, this so-called histogram threshold segmentation. What you do is with a tool, it's really just click on here. It will open for you the threshold tool. With the threshold tool, you can apply a threshold. No threshold means all the values higher than the threshold I set, it will be of interest for me. Okay. In this case, if you like you want to find this object, so these objects are brighter than the background, so you can set an histogram threshold, a gray level threshold with different methods, and then you can extract the contours of it. This is done using analyzed particle. Okay, so here what it really put out for you is the boundaries of your nuclei. Okay, and from there, of course, you can set some measure. But before, I want to show you that when you do automatic stuff, you can have some problems. In this case, for example, one of the main problems you have with this kind of tools of approach, the histogram approach, is the so-called clamped object. So in this case, these are of course two nuclei, or here you have two nuclei even here, and they are not splitted. Another one is the object touching the border. So I will show you very quick, a little bit in the next slides, how to fix these things, okay? The first one is really basic stuff. When you analyze particle, you can ask to exclude object on edges. So this one, if I go back here, these ones here in the new mask are no more here. So you don't see those nuclei because we excluded them. But we still have the clamped object problem. This one, it's a very big field of image analysis, how to declamp split object. One of the main one used is the watershed. Okay, is one of the post-processing. So you have pre-processing like image filtering, like image enhancement, histogram stretching. Then you can do post-processing. I will not really go too much into it. I just want to use some. So for sure we used, for example, on edge exclusion. Now we go to object separation techniques, and one is the watershed. If you apply watershed, what you see is that some of the objects are separated well. Now it works well when you have some object like this. Even here, even now, as you can see, it's just really a button on the Fiji interface. What the watershed suffers on the other side is the so-called oversplitting. If you see, those ones were good nuclei, but then our algorithm was able to recover this one, so to split those one, but this one, it oversplit them. So to overcome this issue, a lot of study in the fields have been done. So you can, of course, think about more elaborated and complicated variation of the watershed algorithm. You can see more preprocessing like filtering, edge detection, or you can go for completely different approaches for splitting objects. Okay, so this one. Okay, from here you can extract feature, as you can see. Yes, so you can extract area, the mean intensity, it's really measure them, okay? You can measure them. What I want to show you also, let me see the time, like 5 minutes, is enough for me. I want to show you that, of course, also the biological image field is now in the machine learning artificial intelligence wave. So there are a lot of cell and nuclei segmentation tools developed with machine learning. And one, for sure, since finding nuclei is a very important, usually biological question. So they developed methods to be able to extract nuclei and, let's say, overcome, do better in that splitting issues we have seen. So I just want to show you that now is integrated in Fiji, so you can have this StarDist algorithm that is one of the most used. And what I want to show you is that without changing any parameters, with situation easy like this, it really it's fast, and then it works quite well. Okay, it has split this one good, it didn't split this one, it didn't split this one. So of course, even these algorithms, they suffer some problems. So here, for example, you see that this one was missing. We still have the touching border object, one here, and so on. And here it was not able to fix this nucleoid. So of course, oversplitting is quite resolved. There are some missing nuclear problems. This is maybe you can fine tune the model, the StarDist parameter, and of course you can, with post-processing, you can easily read out of the touching border object. So we are getting at the end. I just want to show you that this kind of, what we saw today, was just the tip of the iceberg. In Fiji and with Fiji, you can really develop much more complicated tools and analysis. So here I just want to show you some of them. Like this one is it's a tool developed in Fiji, so it's not Python, it's not MATLAB, it's not something outside, it's developed inside Fiji, that it allows you to find the philopodia, so this kind of, how can I say, fingers of the cells, okay, and then it can tell you the tip moving of the cells and something like that. And then you can do nuclei tracking, you can do colocalization analysis, and most of the time these things are just a plugin that you download from the internet, since it's open source. You put inside your Fiji, you try to understand if it works with your environment, and then with your images, with your experiment, and then maybe with just changing some few parameters, you can analyze your image. Okay, so last slide for me. Why Fiji is used, and it's the most used one at the moment. The really main point is that it has an easy graphical user interface. It is for biology, and it's developed for biological background researchers, not for computer scientists at the beginning. At the beginning, it was built for biological users. So they need, even without knowing anything about programming, they really need an easy graphical interface. And this is quite striking is for the field. It was 25 years ago when they introduced it, and it was the first one. And now it's also, because it was the first one, now it's extremely used and still useful. It also includes all the foundational operation of the image analysis. So the image, the point and their filtering, the histogram stretching, the lookup table, all the post-processing, watershed, all this kind of really foundational operation of any image analysis tool. It's developed for biological microscopy data. So when you have multi-parametric acquisition, you have bioformats, you have images coming from a microscope. Fiji already knows that it's you are using it for biological images, so it helps you to interpret them. It includes basic operation, but also more complicated analysis and also machine learning and artificial intelligence. So it's quite powerful. That's what I want to say. It's not only for basic stuff like draw a line and see how long is an object. You can do that. You can visualize an image. You can launch a machine learning algorithms. Then we didn't see it because there is no time, but it's quite easy to write your own Fiji pipeline to automate your analysis. Maybe will be another webinar for sure, not today. Okay, for me, I'm finished. So we thank a lot African bioimaging community. Follow it because it has a lot of webinar, it has a lot of resources, it has a lot of funding suggestion. So it's really important to fund it, to join it if you're an African researcher or an African or a person living in Africa. And then this is just my mail if you want to contact me. Okay, thank you everybody. I see some questions.
Thank you so much, Emmanuel. If
Okay.
anyone has questions, please direct them to Emmanuel in the chat, or you can unmute yourself and ask your questions live.
Okay, I see the part of the presentation. Yes, I think the Precious has already replied to it. Okay, okay.
So the video of this webinar will be made available on the ABIC YouTube page by next week Monday. In the meantime, Emmanuel, while we wait for people to ask some questions, here's one. Is Fiji suitable only for fluorescence microscopy, or can it also be used for histopathological images?
Okay, thank you, Precious, for the question. It is a very good question because it allows me to also, we talked a lot about the pros and the good things of Fiji. This helps me to say something about the bad things or the things where Fiji struggles. Where it struggles a lot is when you have very big images, very in size. When the image is very big and the histopathological slide from patient, for example, can be 20,000 pixel size, there is not the best tool to use. You can use for histopathological in the sense that if the image is small, it's quite small, you can still use it. When the images become very big, there are other tools like Kupat that it was introduced by Sarah McCardle in another community webinar of EBIC that is really developed to do the so-called digital pathology. So when you have really histopathological slides. But yes, you can use it, but up to a certain size of the images. And then I want also to add another problem of Fiji is when you have a lot of data analysis to do. Usually you don't do data analysis with Fiji. You can do in a certain way, but usually you extract tables and then you pass the tables to Python, R, or MATLAB, or Excel, and you are doing the data analysis aside because it's not so able to handle tables and data tables. Okay.
Thank you, Emmanuel. That was quite in depth. Is there any plugins that significantly expand Fiji's capabilities? Like, is there any plugin that if you had to add it would make Fiji work 10 times better? Or is it optimized at its current state without having to add in any plugins? Do you know of them?
No, I mean, the good thing is like, is a little bit, how can I say? It's really project dependent, let's say. The Fiji that you download has really a lot already of very good scientific pipeline and plugin already ready for you to use. But then a lot of time what you see is like, I don't know, you are analyzing mitochondria, you go online and search papers that analyze mitochondria, they have done a Fiji plugin for you that is really dependent on what you are asking. So this is why I was stretching a little bit the biological question when you start to do an analysis, because depending on that, you can really find already many extension of Fiji directly usable for your biological question. But the Fiji you download, as I can say, the 80-85% of stuff you need to do most the biological analysis. Then, of course, if you go to more specific uh, specific question, maybe you have to download something, but it's just really download and add it. It's like 2 clicks, you have you add your plugin. And on the other side, you when you become good, you can also do the opposite. You develop your own plugin. You can put the sharing it with the community, and other people will use it.
That's great. Thank you so much. So there's a question here from Shireen Makti. Thank you, Dr. Emanuel, for the presentation. I really enjoyed it. I have a question. Could we use Fiji to evaluate this spheroid rate format of time lapse and behavior, or are there better software you suggest?
You can do it. We do it in my actually like my research. My group does a lot of spheroid time lapse and something like that, and also cell migration. And Fiji, I mean, it reaches a very good level. You can do most of the things with it. Then, if you are really like, I mean, there are better software, but maybe they are much harder to understand and to use. So Fiji, if you need something already good and your images are already good and the situation is not extremely complex, Fiji is more than enough. Of course, if your situation is a little bit different and more complicated, but it's harder for all the software, not only for Fiji. Okay, I hope I replied, Shrin.
Thank you, Emmanuel. I don't know if there's any other questions. If there are, please feel free to put them in the chat or unmute yourself to ask them directly. And then for someone who's just starting out and wanting to analyze images and they're trying to decide which is the best, why am I using Fiji other than its user interface, is what type of specific question should I be asking in order for Fiji to be the software that I use?
For me, it's really what it goes along. It goes along with the first question you asked me. When you have fluorescence image, moreover, also the other ones, but moreover fluorescence images, also the RGB images, but with fluorescence, it was for it, and you don't have extremely big slides, and Fiji is most of the time the really starting point because it's easy to use and you can do really a lot. When you have big images like the one maybe from digital pathology that they are coming out in the last 5 years, this is why they developed Kupat, and this is also why uh, they try a lot in the last, I don't know, 10 years to put out other tools to do image analysis, but Fiji was always eating all of them and a few people use them. Instead, when Kupat came out, so it is around 5 years ago, more or less, it really found a niche where Fiji was not good. Okay, so and this is why Kupat is also used because it has found a niche that is the one with digital pathology, let's say, to be quite wide of the field. There is Kupat is better, I think. Fiji is more useful when you have smaller images, and it's also still more useful, in my opinion, when you want to do visualization, quick stuff, because you can really open them and visualize them quite easily and fast, and you can do even manual stuff very fast. And there is still very useful, because in 5 minutes you can really watch all your images and see how the experiment went. But I mean, it's the go-to guy for a lot, for many biological image analysis.
Okay, thank you so much for that, Emmanuel. If there's no questions from anyone else, I think we can bring this webinar to a close. So thank you so much for this presentation. We really appreciate your time. I hope we can get you to come in again for the topic that you mentioned at the end there that needs a bit more expanding.
Yeah.
That would be great. And for anyone who's here, please do feel free to follow our social media platforms, visit our website. We put up all types of things, opportunities, events, funding opportunities are shown up there, and also sign up to our mailing list so that you can get our newsletters, where we put up the most recent updates within the community and any opportunities and events that you guys can attend. The recording will go up next week on the ABIC YouTube page, so just keep your eye out for that as well. And follow Emmanuel on his different social media as well, and contact him if you have any questions.